Hello, my wonderful AP Bio students. This is Ms. Hardy, and I just wanted to record a short little review video for you on concept 11.1. I know cellular communication can be tricky and there's a lot of detail, so I just wanted to help you out with a little review video. Before we get into the review, I just want to ask the most important question of this video. You already know what it is.
Who's excited for bio?
Woohoo! I know I am, and again, I hope you find this video helpful. All right, let's get into it. So when we think about cell communication, it has a lot to do with something known as cell signaling, which is when cells are talking to each other, okay, sending signals to each other so they can recognize each other and lead to an action. Well, let's go ahead and start with the basics, a basic example of cell signaling, which is in yeast. Yeast obviously need to be able to identify their mating types in order to reproduce and thus survive. And the way they do that is through cell signaling. For example, in yeast there are type A and type alpha cells, and the type of the cell they are is based off of the type of factor they release. So type A cells are going to secrete factor A, while type alpha cells secrete factor alpha. Now, in order for them to reproduce, they bind to the other type of cell. And for them to recognize that type of cell, they have receptors, okay? So type A is going to have receptors for factor alpha, which is released by type alpha. And type alpha cells will have receptors for factor A, which are released by type A cells. And so in that way, they're able to recognize when the opposite type of cell is nearby, and when they both recognize each other, they're able to bind to each other, create a new A/alpha cell, and then from there, they can start to reproduce. Now, this combination of A and alpha cells allows for evolutionary advantages because they now have two sets of genes, and it allows for diversity and adaptations to environments when they reproduce in this way. Okay, now this is just an example of how yeasts ensure that they accurately pair with the correct mating type before they fuse and then start to reproduce. Now, the binding of the mating factor, when that happens, actually leads to a cellular response of mating through something known as a signal transduction pathway. And we're going to learn a lot more about signal transduction pathways in later concepts, but this is just like a little introduction that there's not just a binding and then everything happens. There is a series of steps that follow the binding of the mating factor to the cell, which thus allows for the mating and the combination of those two cells to one. Now, where did these signal transduction pathways arise? When did that happen? And let's just go ahead and go to the simple organisms again. Let's start with prokaryotes and single-celled eukaryotes. Now, bacterial cells, like prokaryotes, they secrete molecules that can be detected by other bacterial cells. Okay? This is something known as quorum sensing, which is the ability for these bacterial cells to sense the concentration of signaling molecules around them. So if there's more signaling molecules, they know there's a lot of bacteria around them, which will help them monitor local cell density. So again, the more signaling molecules, they're like, oh, I'm surrounded by my buddy bacterial cells. There's a lot of us here. And when there's a lot of them there, it allows for them to create things such as biofilms. So again, when they're all together, they feel that local cell density. They can act together and create things like biofilms, which are when a group of bacterial cells stick to a surface. So one example, forgive me, this is gross, is the plaque on your teeth. That is a biofilm created by bacteria on your teeth adhering to your teeth surface. And so that's why we need to brush our teeth, floss our teeth to get rid of those bacterial cells so they don't harm our teeth, okay? Because what these bacterial cells do when they're in a biofilm is they derive nutrition from that surface. And in some toxic cases of bacteria, they actually secrete toxins and they affect their host. And so we want to be very mindful of these biofilms because they can harm us. And in medicines, it has some implications because if we know that bacteria work in this way to form biofilms, we can disrupt quorum sensing, which will prevent them from forming these biofilms working together in order to, unfortunately, leech off of their host. In this picture, we have another example of bacterial communication in which these individual rod-shaped cells are known as slime bacteria. And what they can do is when nutrients are not as available, they actually aggregate into these spores or these fruiting bodies that are capable of surviving until nutrient availability improves or conditions improve. And so in sum, from this slide, you can tell that communicating with other cells allows for these bacteria to survive better, right? So whether it's quorum sensing, where they can start to gather nutrients for their own benefit, or whether it's in the formation of these fooding bodies where they can survive until conditions improve. So we've talked a lot about simple organisms like bacteria, like yeast, but what about communication in multicellular organisms when there's multiple cells coming together to work for the betterment of one organism? There's a couple types of communication that occurs. So the first type, we're going to go from closest to furthest. The first type is direct contact. So based off the name, you can tell these cells are in direct contact. They are touching. And so in class we mentioned juxtacrine signaling. This is juxtacrine signaling, right? They're very close together. Another name is just direct contact. We've learned about these two types of direct contact. There's gap junctions in animal cells and plasmodesmata in plant cells. They're just little channels or holes that allow for passage of molecules. So for example, in gap junctions, it allows for nucleotides, sugars, amino acids, and ions to pass through. In plasmodesmata, proteins, ions, water, and chemical signals can pass through. There's also cell-to-cell recognition, where instead of having kind of junctions, openings in the membranes or in the cell walls of these cells, there's interaction between the molecules on the surface of the cell. So you can think about the carbohydrates on the surface of cells, the glycoproteins. Those allow for cell-to-cell recognition to occur. So let's take a step a little bit further. Instead of the cells directly touching, they're a little further away, but they're still nearby. And this is local signaling. And so in local signaling, there are signaling molecules that are secreted by a signaling cell, which is known as the local regulator. Okay, so they release these chemicals which are received by target cells nearby. Now, one example of local signaling is paracrine signaling, and that is just the general term in which local regulator cells are going to influence cells that are close by. And an example of a molecule that's released or a signal that's released are growth factors. Another type of local signaling is synaptic signaling, and this is local signaling specific to the nervous system. So it happens between neurons or nerve cells. Okay, and what happens here is the axon terminal, that's this region over here of one nerve cell, is going to release these chemicals known as neurotransmitters across something known as a synapse. So this space between these two cells is known as a synapse. And so these neurotransmitters are going to diffuse across the synapse, reach the target cell where there are receptors there waiting to receive the signal. And we're going to step a little bit further away. Now there's long distance signaling. And so in long distance signaling, this is known as endocrine signaling, and there's the use of hormones. Specialized cells release these hormones. These are still chemicals. And what these hormones are going to do is they're going to travel through the circulatory system far, far away to a target organ and eventually a target cell. So endocrine signaling is what we call it when it happens in animal cells, but there's also something known as plant growth regulators, and these are hormones in plants. Okay, and so plant growth regulators, they don't travel as much through like vessels, like liquid vessels in plants, but they often move through cells or diffuse in the air outside the cells. So hormones can come in different sizes and types, and we'll use one plant hormone and one animal hormone to demonstrate this. In plants, there's ethylene. And so if you've ever had like a bunch of bananas and they ripen really quickly when they're together, it's because of ethylene. Ethylene is a gas that promotes fruit ripening and regulates plant growth. And so if you look at the molecular formula of ethylene, it is very small, right? It is only C2H4, so it's 6 atoms. It's a hydrophobic molecule. It's small enough to pass through cell walls, pass through cell membranes. And so that's ethylene, the structure of ethylene. Again, here's a little pro tip. If you've ever had bananas and you want them to last longer before they all turn brown, then you can tear them apart from their bunch and then leave them far away from each other, because the ethylene that's released will influence the other bananas and cause them all to ripen really quickly. But if you're trying to make banana bread, then keep them together in a bunch. Maybe you can put them in a paper bag, and that's going to allow for the concentration of ethylene to be greater and promote more fruit ripening and happen a lot quicker. A little fun example for you. So in animal cells, there's insulin. This is a hormone that's going to regulate sugar levels in blood. And this is a protein that's made of thousands of atoms. So as you can see here, they're different sizes and they have different roles as well. So now we've talked about different ways cells communicate with each other and signal to each other. What happens when the target cell is exposed to the signaling molecule? How does a response come about? And this is something that was explored by Sutherland. And so Sutherland ran an experiment and he focused on epinephrine, which is a hormone that triggers the fight or flight response. So if you ever think like adrenaline rush, it's kind of like that. That's what epinephrine is doing for us. what it's actually doing in our body is it's simulating the breakdown of glycogen, and when glycogen is broken down, it releases glucose. And so this really happens in our liver because glycogen is found a lot in our livers. And so our liver or muscle cell can use the glucose that's released for energy production. Now we need the energy because when you're in the fight or flight response, you're faced with a very scary stimulus, your survival is on the line, and you need energy to either defend yourself or to run away from that threat. So Sutherland ran his experiment, and what he discovered, what his takeaway was that a pathway leads to the response of glycogen breakdown. It's not just that there's one enzyme in isolation that is activated by epinephrine, but there's a pathway that leads to the activation of the enzyme and thus the breakdown of glycogen. And how he basically tested this out was he ran 2 experiments, 2 conditions. In one condition, he had a beaker and he had epinephrine plus the enzyme, just in isolation, just those 2 things mixed together. And when he put those together, he noticed that there was no breakdown of glycogen. So what that shows us is that epinephrine and the enzyme do not bind directly to each other. They don't interact directly. If they did, then there would be a breakdown, but they didn't interact directly, and so there was no breakdown. When he put epinephrine plus enzyme in an intact cell, there was then a breakdown of glycogen into glucose. And so what this shows us is that the cell must provide some additional intermediates that allow for the breakdown of glycogen that would not otherwise be present if there was no intact cell. It also shows the importance of an intact membrane on a cell that then allows for the transduction of a signal. And so with this information, he was able to start to give us clues about the signal transduction pathway that exists inside of our cells. So now let's go ahead and talk a little bit more about what this pathway is and what happens in this signal transduction pathway. There's 3 main steps, starting with reception. So this is just the detection of the ligand. As you can see here, the signaling molecule is binding to receptor. They are specific in shape, and so only one ligand will bind to one receptor that can lead to a very specific response. After reception occurs, we have transduction, and that's a conversion of the signal. And as you can see, there are usually multiple steps before it gets to the final response in the cell. And so there's multiple relay molecules, multiple intermediates that basically play a role in the transduction or the conversion of the signal. And this plays a big role in amplification of the signal as well, allowing for only one binding of one signaling molecule to one receptor to a really big response inside of the cell. And finally, of course, the whole purpose of a signal transduction pathway is a specific response from the cell. And so we go from reception to transduction to response. And this can be a variety of responses. It can be catalysis by an enzyme. It can be rearrangement of the cytoskeleton. It can be activation of genes, production of proteins. In short, these types of pathways allow for normal functioning of an organism. All right, so in summary, here are all the things you should have learned about and reviewed in this video. First thing is the evolution of cell signaling. We talked about the yeast and their mating factors and how that was a simple example of how different cells can recognize each other and lead to a response. We then learned about bacteria and how they sense the presence of other bacteria around them with quorum sensing and biofilms that they create. We also talked about the different types of communication that exist inside of multicellular organisms. So that's direct contact communication. There's also local versus long distance signaling. We talked about examples of these types of local versus long distance signaling. So there's paracrine signaling, which has to do with growth factors as an example. There's synaptic signaling, which has to do with neurotransmitters being released. And then endocrine, which is long distance signaling, has to do with hormones being released. And finally, we discussed the stages of cell signaling. We learned that cell signaling often is not just the mixture of an enzyme and a ligand, but rather that there are many steps in between, and it takes place in a signal transduction pathway. And so there's 3 stages: reception, transduction, and response. I hope you found this video helpful, and always remember, this is fine. I can do it. I'll see you in class. Bye!